The challenge today is to demonstrate that one, evolution is scientific, and two, that it can account for the liver coming from what we would reasonably consider a non-liver. To demonstrate the former, first I'll distinguish between what counts as scientific evidence and what does not. A common misuse of the term comes from people taking an observation which their model happens to be able to accommodate, and taking this to be evidence of the model itself. For example, people will often point to a fossil which appears to be halfway between two existing animals and take this to be evidence of common descent from a transitional species, when in reality, this is in fact a post hoc rationalization. You are asserting a cause when you do this. You need evidence for your explanation, not an explanation for your evidence. So, what is the proper way to argue this? And how is evolution a valid science despite the epistemic polluters who often argue for it? First, let's give evolution, by which I mean the process and not the theory, a definition. A simple definition that most people can agree on is descent with modification. We observe that replication entails four properties, which I have enumerated on the screen. They are one, that replication entails inherent variance such that individuals are unique. Two, traits are transferred vertically via inheritance. Three, unique inherent traits differ in the qualities attributable to their fecundity. And four, there is an inequality in the frequency with which the inherent variable traits survive and reproduce. This, and nothing else, not fossils, not homologies, and not genetics, serves as the observational basis for our theory. What I have just described is an observed mechanism for biological variable change, and it is this mechanism which I claim can account for the diversity of life and the differences between any two inherent traits. This is the central hypothesis on which the theory is based. Let's now apply this mechanism and a proper understanding of standard of evidence to the evolution of the liver from a non-liver. In order to do this, let's evaluate the claim. This evolution actually presupposes a non-evidentiary claim, which first needs to be justified. We need to demonstrate that certain conditions can be met. The first condition is that the explanation cannot allow for the interference from an entity other than the mechanism. The evolutionary transitions must be incremental and within the bounds of observed sources of variation. Each increment must have a selective quality, or in other words, we can't have gaps where one increment appears to have no reason to exist other than to build the next increment. This non-evidentiary claim is in the form of, if the observed mechanism meets these conditions, then it can account for the liver. And by the way, because this has to be said, I will prove this, but once I do, this will in no way obligate me to presuppose a natural explanation for anything prior to the non-liver. The claim would be factual regardless, and you would be stripped of this as an example of a valid argument against evolution. My argument does not need such an assumption to be granted in order to be valid. With that out of the way, what does the liver do, and what are the steps to evolve it? The liver has several functions, including metabolic functions, detoxification functions, digestive functions, protein synthesis, and more. On account of these functions, I want you to consider a microscopic animal. And in case you're wondering, yes, those exist, where each individual cell in its body will be far more impactful than in a larger organism. These animals obviously have relatively low complexity due to having so few cells. For example, functions like breathing, waste removal, detoxification, and even digestion can be performed by the skin or by just one or a few cells in these animals. The point of this is there doesn't exist a function of the liver which cannot be performed at the cellular level. And I think we would agree that a skin cell which performs detoxification is a non-liver. The question then is how to go from this arrangement to one where these cells are organized into tissues, which themselves are connected by ducts to other tissue systems. And we need to accomplish this without violating any of the conditions we imposed earlier. The logical next step would be the connecting of the duct system, because if the complexity of the tissue increased first, this would represent an increment without a selective reason. By depositing material such as bile into the gut rather than wasting it through the skin, it is recycled to increase the efficiency of digestion and the breakdown of lipids, thus conferring selection. This increment is actually simpler than it seems. If the cells in the animal are already porous such that waste material is transported to the skin, where it is secreted, a number of observed sources of variation can account for the formation of a channel into the gut rather than the skin. For example, a duplication of a cell or cells can create the space for some to exit through the skin, while others exit into the gut. Or a cell may inherit a cylindrical shape, or a new orientation, which deposits material to the gut that way. Even a single cell in this animal may itself function as a duct into the gut. There's even a word for this cell. They are called flame cells or tube cells. Here we have the same information rearranged and slightly modified to a new function. Is this a liver? Having implemented a duct from a non-duct according to the conditions we imposed, we can solve the next increment, which is the compartmentalization of the cells attached to this transport system. This simply involves connecting cells which already perform functions now associated with the liver to each other and to the new gut complex in the same manner as before and to the same selective effect. Upon this increment, a duplication of cells lining the gut would have the effect of increasing its surface area and thus storage capacity for sugars, proteins, and enzymes deposited by the complex. Just as a channel can be formed into the gut, so too can one deposit toxic material from the gut to this cell complex. Detoxification functions are still performed at the cellular level, but now we're concentrated at the site of the complex. Is this a liver? At this stage, I have represented the bulk of the necessary increments. Such an animal has no need for a circulatory system, or even blood for that matter, as all the oxygen it needs can be passively diffused from the water through the skin to its cells. At this level of complexity, it may now finally be just large enough to be visible to the naked eye. But in order to get any bigger and adopt some of the greater complexities of a true liver, a circulatory system will be required. And that will have to be its own video, unfortunately. Hopefully by now I've provided enough argument for people to chew on and discuss up to this point before I argue the remainder of the complexity via these criteria. For the sake of brevity, I apologize that I'm going to breeze through this part. I'll just leave you with some context on what the simplest, quote, circulatory system entails. I've repeatedly referred to the gut, which refers to the endoderm, or in other words, the innermost cavity of the body, which is the first to embryologically develop. The circulatory system, however, is developed from the mesoderm, or middle layer between the gut and the epidermis. In this animal, the mesoderm forms a fluid-filled body cavity where transport is facilitated by ciliated cells rather than a heart-like pumping system. There is a bath of free-moving organs and possibly blood cells rather than a true vascular system. This setup is well documented by living examples. One of the original claims of this video was that the evolutionary mechanism can account for the diversity of life and the differences between any two inherent traits. This is the evidentiary claim I am building a case to. Thus far, I have demonstrated that it can account for the difference between what is decidedly a non-liver and an intermediate. As our study of biology becomes more advanced and new discoveries get more granular, as we increase the opportunities to find ways to falsify worldwide diversification via descent with modification, we find that of all the examples that occur in nature, we don't find a single one which cannot be reduced to this process. I don't know about you, but I call that a powerful theory. I'll continue to argue this for the remainder of the liver complexity next time.